In this video, we will discuss about the final figure of this paper, where they talk about the validation of mKRAS specific T cells from vaccinated patients PBMCs using single-cell RNA-seq data, and they also check whether this TCR or mKRAS specific T cells are able to detect the tumor cells and can differentiate between the tumor cells and healthy cells or not. So first, they started with this UMAP for this PBMCs before and after these mKRAS vaccinations, and they are able to find out that in the UMAP, the most of the T cells post vaccinated belongs to the CD4 plus, whereas very few CD8 plus populations are present, and this is in line with their previous results, where they have found out from the mass spectrometry also that CD4 plus cells are high and CD8 plus cells are low in their mKRAS vaccinated PBMCs, and they use single cell to also classify the individual T cells in the PBMCs that are exposed to individual mKRAS antigens. Similar experiment they did using mass spectrometry in the previous studies, in the previous experiment that we have discussed, and here they did it using the single cell RNA-seq data or identifying the T cell receptor sequencing, and similar to the results that they seen from the UMAP, here also they find out that CD4 plus positive cells are high compared to the CD8 plus positive cells, and they also see some of the other T cells also, but those roles they have not mentioned in this figure. So after validating that using the single cell RNA-seq data that there are T cell receptors present in the post vaccinated PBMCs which are exposed to the mKRAS antigen or mKRAS peptides, now they wanted to design an assay to check how specific are these T cell receptors. So what they did is they take the Jurkat cells, or more specifically they take this TCR knockout Jurkat cells, and that also has this NFAT GFP reporter, and so NFAT GFP reporter will give GFP signals whenever these Jurkat cells are activated, and they activated when they successfully able to interact with the tumor cells. So they took these TCR knockout Jurkat cells and then transduced them with individual TCR specific peptide, and then co-culture them with these PBMCs, and then they find out the GFP, quantify the GFP signal using flow cytometer. So for this mono-reactive TCR, they are able to see the signals only for the individual. So they co-culture them not with the PBMCs but with this patient matched lymphoblast cell lines. So the results that they've seen is that the GFP signals are specific for this mono TCR receptors. They are able to see signals only for the individual mutations of the mKRAS, and for this cross-reactive they are able to see signals for this all the or most of the mKRAS mutations, but the signal intensities are weak compared to the monotherapies. Specificity in the mono reactive TCR. Then for one of this cross-reactive, they shown the quantification results, where they can see that it is reactive to the different mKRAS, different mutations of this KRAS gene. And then they want to check what actually helps in this TCR interactions with this lymphoblast in this case. So they use these three different surface receptors, that is HLA-DR, HLA-DP and HLA-DQ, and they are able to find, and what it is they took this K562 cell lines and transduced them with these three surface receptors individually, and they can see that TCR specificity signals are high when they have these HLA-DR overexpressed, but not with the other conditions. However, when they check the patient only have this allele, that is DQB. So in the patient they check it with the donor that have DQB, and then donor does not have DQB, and they can find out that when the donors has the DQB, the specificities are high. That is, they check it for the G13D, but when the same patient's DOP has been knocked down, the specificities are low. Indicate that this cell surface markers also, there is no marker, the cell surface receptors, that is DQB in this case, also plays a role towards this TCR detection, also plays a role towards the TCR interactions with this tumor cells or lymphoblast cell lines in this case. And they also check for the public TCR β08 in the Jurkat cells, and in this case they are able to find out that it can give signals for the different mutations, different for all the patients for the mutated mKRAS, which has this mutated mKRAS. But when they use the wild type or the control with the irrelevant peptide, they do not give any signals. So let me reiterate it. So what they did is they co-culture these Jurkat cells with TCR β08 along with this lymphoblast cell lines of the patient and with different mKRAS, individual mKRAS peptide. So when they use the G13D, they are able to see signals, and when they don't use the G13D or they use G13D or they use the wild type mKRAS peptide, they do not see signals. So this is true for all the three cases that they have checked. So this indicates the further specificities of their TCR. Now so far they checked it on the lymphoblast cell line. Now they wanted to check whether it can interact with the actual tumors or not. So for that they took this CAPAN-1 that has this KRAS G12V, and for the control they used SUDHL 10, which has the KRAS wild type, and they are able to see that when this CAPAN-1 KRAS G12V co-cultured with the Jurkat cells, and Jurkat cells condition are same that they have used in the panel C. The Jurkat cells that have this NFAT GFP reporter, and they are transduced with individual TCR that is specific to the mono-reactive TCR specific to the particular mutations. So in this CAPAN-1 case it is specific to the G12V mutations. So when these cell lines are or when these Jurkat cells and CAPAN cells are not co-cultured along with this peptide or along with this mKRAS G12V peptide for the CAPAN-1, they do not see any signals. And similar results they have seen for this MDA-MB-231, where G13D peptide is not co-cultured with Jurkat cells and the tumor cells. They do not see activations or detections. But when they treat them with individual with their specific mutated peptide, that is in the G12V case, they are able to see the G12V signals, and for wild type the signals are low, but and for the G12A case also they are able to see signals, and here the wild type signals are also high. For G12C there are no signals, the wild type signals are all there to some extent, and for G13D there are no signals. For wild type there are some signals. However, we can see that intensities of the signals for this G12V in the CAPAN-1 case compared to the SUDHL case is significant, but they do not shows the triplicate results that have the statistical analysis in this figure. Maybe we need to check in the supplemental figures, they might have shown it there. However, we can clearly see the difference between the CAPAN-1 cells and wild type cells. And similarly for this MDA-B3 also, when they co-cultured them with this KRAS G13D peptide, they are able to see signals. So this all indicates that This all indicates the validation of this mKRAS specific T cells and its preferential selection towards mutated mKRAS compared to the wild type KRAS. So I hope you understood how they validate their mKRAS specific T cells and how they check them in the whether they are how they check its specificity on the tumor cells that has the mutated mKRAS or the wild type mKRAS. That's all for this video. If you need to understand more about this paper in detail, you can go through the reference that has been provided in the description. Thank you for your time.